Good morning, everyone. I'm Dr. Preeti Sharma. Today I'll be speaking on various cellular organelles and different types of cells. Cell, as we all know, it is a structural and functional unit of the life. It is the basic unit of biological activity. The cells of the biological systems are divided into the two categories. One is the prokaryotic and eukaryotic. This is the very advanced version of cell, that is a eukaryotic cell. Let us see the differences between the prokaryotic and your eukaryotic cells. Prokaryotic means a very primitive kind of cell. Once the organic evolution happens, the first kind of cells which came into the appearance, they were the prokaryotic cell. In course of time with evolution, lot of changes happened into the prokaryotic cells and it converted into the eukaryotic cells. So there are a lot of differences with respect to the size, cell membrane, cytoplasm. So you can see it here, the size is very less, that is 0.1 to 10 micrometer, and the eukaryotic it is 10 to 100 micrometer. The cell membrane is a very rigid cell wall in the prokaryotics, while it is a proper cell membrane. Then cytoplasm is viscous with no subcellular organelles. Subcellular organelles are present into the eukaryotic cells. Then about the different organelles present. So the nucleus in the prokaryotic cells, the nuclear zone with DNA is available. It is not very well defined, while in the eukaryotes it is well defined. The mitochondria are absent in the prokaryotes. Golgi apparatus, cytoskeletons, they are absent. While they are present in the eukaryotes, then we talk about the cell division. Cell division happens through fission, while in the eukaryotes it happens through mitosis. So you can see the structure of prokaryotic and eukaryotic cells. The various line of differences is apparent from the structure itself. Well-defined plasma membrane is here in the eukaryotic cell, while it is bounded by bounded by cell wall as well. It is bounded by cell wall. Then cytoplasm is there. DNA is in the cytoplasm, while the DNA here is within the nucleus, bounded by the nuclear membrane. Ribosome is the only structure available here. Rest of the membranous subcellular organelles are absent into the eukaryotic cell. Now let us have look onto the separation of different cellular organelles while for study purpose. So first of all, a suspension is being made by homogenizing the tissue fraction. The suspension of broken cell contains the subcellular components such as lysosome, peroxisome, and membrane fragments. And this suspension is being centrifuged for 10 minutes at 800 gravitational pull, followed by the centrifugation of the supernatant at 15,000 gravitational pull for 10 minutes is being performed. So in this way, we can separate the nuclear sediments which are lying under the bottom. Then in the next step, the centrifuge supernatant is being centrifugated at 1 lakh gravitational pull for 60 minutes. So rest of the cellular organelles, including the mitochondria, lysosomes, and peroxisome, they sediments down the bottom. In the next step, the centrifuge supernatant is made to undergo 2 lakhs gravitational flow centrifugation for 3 hours. So fragments of the plasma membrane and endoplasmic reticulum segments, they are all sedimenting down the bottom of the test tube. Then in the next step, supernatant contain the we find the supernatant and the sediment in the last, which contains the ribosomal sediments. So the following organelles we are going to discuss here: the nucleus, the endoplasmic reticulum, the Golgi apparatus, the mitochondria, the lysosome, and the peroxisome. Organelles have very specific function. Their specific enzyme activities serve as a marker of organelles during isolation. This is the structure of nucleus of the eukaryotic cell. You can see it here. It is bounded by a nuclear envelope or nuclear membrane. Within the nucleus, there is a very well marked nucleolus. Darkly stained region is there. Then, in continuation with the wall of this nuclear membrane, there is a endoplasmic reticulum. This is endoplasmic reticulum here. And then ribosomes are sticking to the endoplasmic reticulum. And this is internally, this nucleus contains lot of chromosomes. We all know this is the structure of chromosome. Chromosome does consist of DNA. So DNA is here in a very organized manner. It is wounded here. Like you can see it, these are the chromatin fibers, 30 nanometer in diameter, and how nicely they are bounded in the form of nucleosome with the help of histone protein. So histone proteins are helping to bind in a plate-like manners, and this is how in the chromosomes the different chromosomes, the nucleosomes are arranged. So let us talk on endoplasmic reticulum. You can see the structure here: network of interconnecting membrane that form a thread-like structure, and the nuclear envelope is in continuation with the endoplasmic reticulum. It can be of two types: smooth endoplasmic reticulum and rough endoplasmic reticulum, that is also known as ergastoplasm. Then Golgi apparatus is a network of flattened smooth membrane sacs-like structures are available. They are known as sterni. So you can see it here. In continuation with the endoplasmic reticulum, these are the folded structures. These structures are nothing but they are Golgi apparatus, and they consist of lot many sac-like structures. They are known as cisternae. The marker enzyme is galactosyl transferase. Then move on to the lysosome. It is a vesicular structure. The size is 0.4 micrometers, surrounded by the lipoprotein membrane. It is high, acidic. pH is around 5. It contains a group of hydrolytic enzyme. More than 40 hydrolytic enzymes are available in this. Then marker enzyme is acid phosphatase. So lysosome recycle material by breaking down the worn out parts of the cells into the smaller unit. They deliver this material to the cytoplasm for use in constructing new proteins. So if the membrane of the cytoplasm of the lysosome breaks down, it becomes a very, very lethal condition. The enzymes released may also destroy the cell itself. So giving the lysosome the name as a suicidal bag. Cellular digestion, they have important role to play in cellular digestion. Hydrolysis of all junk carbohydrates in the form of glycoside, fucosides, hyaluronidase, then proteins in the form of cathepsin, collagenase, elastase, lipids, so many kinds of lipids, fatty acyltrace, phospholipase, and nucleic acid. All these substances are being digested with the help of these hydrolytic enzymes present into the lysosome. Then let us look onto the clinical application. Clinically, this organelle is very important. Inclusion cell disease. It is a kind of protein targeting defect. It leads to the accumulation of ungraded molecules like inclusion bodies. Second important application is in Important clinical application is in gout. So in this particular disease, what happened? The urate crystals are phagocytosed and cause the physical damage to the lysosome, releasing the enzyme, producing the inflammation and arthritis. Cathepsins are implicated in tumor metastasis. They degrade the basal lamina of the cell. Silicosis is another disorder concerned with the lysosomal disorder. So lysosomal enzymes are released, stimulating the fibroblast proliferation. The diverse group of organelles found in the eukaryotic cells, in most of the human cells, their abundance ranges from less than 100 to more than 100 thousands per cell. So important role which is being played by the peroxisome. It is the metabolism of free oxygen radicals, synthesis of cholesterol and ether lipids. Then bile acid formation also takes place in the peroxisome. Catabolism of long chain fatty acid, catabolism of purine, prostaglandin, leukotrienes all takes place in the peroxisome. Alcohol detoxification in liver is also concerned with the peroxisome. Then metabolism of estradiol. So all these important roles are being played by the microsomes. They are named so because they are unique. They play a very important role in hydrogen peroxide metabolism. These two reactions are very, very important. Have a look on this. The certain enzyme, they contains the enzyme that use the molecular oxygen to remove hydrogen atom from the specific substrates. You can look it here. This is any substrate. With the help of oxygen, it's being converted into H2O2. So hydrogen peroxide is used up and detoxified. As we know, it is a very, very active chemical, converts to hydroxy free radical and starts a chain or body of reactions leading to the damage of various cellular organelles. So the enzyme catalase in the peroxisome converts this hydrogen peroxide to water. This is how it saves the cell from the lethal effect of H2O2. Then have a look on peroxisomal biogenesis disorder. Deficiency of peroxisomal matrix protein can lead to adenoleukodystrophy, or that is also known as Brown-Schilder's disease, characterized by progressive degeneration of liver, kidney, and brain. It is a rare autosomal recessive condition. The defect is due to insufficient oxidation of very long chain fatty acids by peroxisome. Second important disorder is Zellweger syndrome. The proteins are not transported into the peroxisome. This leads to the formation of empty peroxisome or peroxisomal ghost inside the cell. The third disorder is primary hyperoxaluria, is due to the defective peroxisomal metabolism of glyoxylate derived from glycine. Now, the next organelle is the mitochondria. It is a spherical, oval, or rod-like structure. Size is 0.2 to 0.8 micrometer. The number of mitochondria in a cell varies. Erythrocytes, they don't have mitochondria. The liver cells contain around 800 to 2500 mitochondria per cell. Hepatocytes, then it is a bilayered structure. You can see it here. Two layers are easily, you can see it here. Outer membrane, then inner membrane. So outer one is the smooth one, while the inner is convoluted in the form of folds. The purpose is to increase the surface area. So these convolutions are known as crista actually, and the two compartments, are there. One is because of this kind of texture, it has two compartments. One is the intermembrane space, and the second is the matrix part. You can see it here in the middle. There is a matrix part. Then intramolecular inner mitochondrial membrane. This membrane contains the enzyme of electron transport chain, while the matrix contain the enzymes of tricarboxylic acid cycle or Krebs cycle, and the beta oxidation of fatty acid. Okay. So it has important role to play in production of ATP, concerned with cellular respiration, oxidation of carbohydrate and lipids. You have seen lot many enzymes are present. Because of them, carbohydrates and um, proteins can be nicely digested, can be digested completely. Then carbohydrate and lipids actually. Then it is also concerned with the synthesis of urea partially, and partially it is also few reactions of heme synthesis happen in the mitochondria. Now have a look on clinical aspects. Lump's disease, defective energy transduction, then mitochondrial myopathies. It is a Oxford disease due to the mutations in the mitochondrial DNA, because mitochondria has its own DNA. Parkinson's, cardiomyopathies, the age-related degenerative diseases. Antibiotics inhibiting the bacterial protein synthesis do not affect the cellular processes but inhibit the mitochondrial protein synthesis. Mitochondria is considered parasites which entered the cell during the course of evolution. So this is the myological membrane. Next is biological membrane. It is a trilaminar structure, consists of lipid, phospholipid protein, phospholipid in continuation with this 50 to 80 angstrom. It is slightly dynamic structure. It is amphipathic. The lipid contents are around 50%. 50% is protein, and rest remaining traces amount carbohydrate is present. This is a double layered structure. You can see it here. So upper side, these globular structures are, these are the lipid head, phospholipid heads are there, both side, outer side and towards the inside. And these are the long chain of fatty acids arranged against each other from both the layers. Then these are the proteins. They are poked at different intervals within uh, the layer of this lipids. So the membranes are asymmetrical, irregular distribution of protein is here, and proteins are arranged in various forms in the form of channel, peripheral protein, integral protein, that I'll be discussing in detail when I'll be talking on cell membrane transport. Carbohydrates are located onto the external surface in the form of calyx. You can see it here, small sapling-like structures. These are the glycoproteins. See it here. The carbohydrates are then specific enzymes are located exclusively onto the outside and inside of the membrane.